Well, thanks for staying awake and with us this whole time. This has been a lot of talks. It's been a lot of effort on your part, and I appreciate it. John gave you an amazing talk, and he is really a pioneer in that field, and I agree with him that the combination of stem cell therapy and activity-based rehab is really an incredibly powerful tool that is coming. And so please come to this thing at 5:30. It's just to see it, and just to see how the concepts have changed, and those concepts have changed really led by John. I would say, for those of you on baclofen, don't stop it, right? I think that's very intriguing data, but if you stop your baclofen, you're going to have seizures, and that's bad, right? So think about these issues. And I think it's important to think about what we're treating with people on baclofen, but I think some people do need it, and I think you need to work with your physician at that. So let's talk a little bit about stem cells and our approach to treating neurologic disease and where I think this might be going. So we must start, of course, with a talk about embryonic stem cells and where they come from. They come from an in vitro fertilization in a dish in which an oocyte or egg, which had been previously harvested from a woman, is combined with a sperm, and then you form what's called a blastocyst. And a blastocyst is basically a small structure with a few cells on the inside of that, which is called an inner cell mass, which are embryonic stem cells. This is where these very powerful cells come from. From a blastocyst, there are no organs, no tissues, no beating heart, no fingers, nothing which many of us would associate with life. What normally happens is that many fertilizations are done, and 2 or 3 are implanted back into the woman's uterus so that she can have 1 or 2 children. The rest, typically those not implanted because they didn't look right or there was something odd about them, are frozen in liquid nitrogen or are discarded. Those embryonic stem cells are the ones that we would like to study. They are very powerful. They represent great hope, and the bottom line of what I'll talk about is they can do some things that other stem cells can't do. Now, one of the other things that you've heard about in these debates is that, well, adult stem cells have been curing people for decades, whereas embryonic stem cells have not cured anybody yet. Well, part of that's true. Embryonic stem cells have not cured anybody yet. Adult stem cells can be effective for certain diseases. These are stem cells harvested from the bone marrow or from the blood or from the muscle or from the cartilage, and they can be very effective in certain things. Those stem cells can turn into cartilage, blood, muscle. And so for diseases of those, adult stem cells would be potentially just fine. But they cannot do the very specialized cell types in many neurologic diseases. Now, part of the reason why an embryonic stem cell has not yet cured any disease is because we haven't known how to control them. Embryonic stem cells can turn into any cell type in the body, but until about 5 years ago, we haven't known how to direct that process. It's been a random process. And so you know that cartilage cells could develop, you know that muscle cells could develop, but you couldn't control how they developed and how frequently they developed. So a major advance really is 4 or 5 years ago, in which scientists would learn how to specifically create, for example, spinal motor neurons and generate millions of them. And so that's a very important advance, and the way that that's done is that you apply chemicals or growth factors to these embryonic stem cells, specifically directing them down one particular path and avoiding those other paths, which might be cartilage cells or muscle cells, etc. Now, before we get to transplantation, every one of us has endogenous stem cells, and that's part of what John has been talking about, is really harnessing your own regenerative capacity, part of which is with the endogenous stem cells. And I won't talk about the details of this slide, but it is to say that in animals and in humans at all stages of life, in the brain and in the spinal cord, there are endogenous stem cells. These are stem cells primed to repair a tissue after an injury. In other tissues, such as the liver or the gastrointestinal tract, they do exactly that. They repair a tissue after injury. In the nervous system, they don't, to a large degree. They respond to injury, they proliferate, and they make an attempt to repair a tissue, but usually that attempt fails and you are left with a permanent injury. So there's a lot of research going on currently to understand why that occurs. And John makes a very important point. It's a very provocative point that what we know is that in the spinal cord, no new neurons are born after an injury, whether that's transverse myelitis or a spinal cord injury. And he makes the point that with activity-based rehab, you may be able to cause those endogenous stem cells to turn into neurons. And if so, that's a very exciting finding. Here is just one example, and I'll just draw your attention to the cartoon. This is an animal model of a stroke. So a stroke is a blood clot that deprives part of the brain of oxygen. And what you're looking at is a particular area here called the cortex. And these stem cells are around the ventricle in the brain. And in response to injury, what happens to those endogenous stem cells? We're not talking about transplantation here. These stem cells proliferate, they become much more prevalent, and they migrate to the site specifically of injury. So they are sensing something about the injury, proliferating in response to it, migrating in response to it, and attempt repair. So that's very encouraging. It tells us that if we can harness that potential, we could get some significant benefit, even though, as is, that process typically fails and somebody's left with their disability. Now, what about transplantation? So John has led the way, and others have. More recently, Hans Kierstead and colleagues have said, well, okay, so the endogenous stem cells don't work. Let's transplant embryonic stem cell derivatives. In other words, let's do exactly what I was talking about. You take an embryonic stem cell and you specifically direct it to be a certain cell type. In some cases, oligodendrocytes. You've heard about oligodendrocytes, right? They remyelinate the naked axons. So in a demyelinating disorder, if the endogenous stem cells won't do it, what about transplanted cells? And if you can make oligodendrocytes or oligodendrocyte precursors and transplant them directly into a demyelinated area, could they potentially exert a beneficial effect? Well, this is happening, and now the ability to efficiently generate oligodendrocytes has led the way to the planning of human clinical trials. So you'll hear about this. Hans Kierstead and his colleagues and Giron have announced that they will proceed in traumatic spinal cord injury with human embryonic stem cell-derived oligodendrocytes, transplantation. Now, I don't know when that's going to happen. They suggest that it will happen within a year. I think it's not going to occur that rapidly. I think it's going to be a couple years away. I will tell you, though, that we, for example, are considering a transplantation of GRPs, glial-restricted precursors. These are human glial-restricted precursors, and there is a company that has developed these cells and has decided that the optimal market, the optimal test group for these cells and whether they myelinate the demyelinated nervous system is transverse myelitis. And so Chitra is running that trial. Chitra, raise your hand. You all know who she is. And so she's gathering all the details, and it takes a lot of work, and now that she's done with this symposium, that's what we're hoping that she will do, is to shift gears and to say, listen, this is an absolute priority to ask patients to participate if they're interested. Patients with a stable, not acute, stable, but incomplete transverse myelitis with a clearly defined lesion in which we can test the transplantation of glial-restricted precursors, and we will look for a functional recovery, we will look for an MRI recovery with new imaging modalities, and we will do a physiologic or electrical recovery after transplantation. Details to come, but that's in the works. Now, when we think about transplantation, I've told you about one of the cell types, an oligodendrocyte. And so you can say, I want to make oligodendrocytes, and I want to transplant them in a demyelinating disorder. Or you can think about neurons, or you could think about astrocytes, which are the support cells present within the nervous system. You can then transplant them and think about a variety of readouts. Restoration of a neural circuit. John put that at the very top of his list, and very appropriately so. It's a very complicated task, is to rewire the nervous system, but there are other things that you could do with the stem cells. What if the stem cells secreted growth factors and augmented the function of the host spinal cord? Those types of things could all be used as a therapy in neurologic diseases, including motor neuron diseases. Now, do I have to do anything? I want to show you a cartoon. Now, everybody met Ben Greenberg, right? You're going to hear his voice.
The ability to move requires that the brain and spinal cord communicate with muscles. This is done through special neural circuits made up of nerve cells called neurons and their long wires called axons. In the analogy of an electrical circuit in a house, turning on a light switch starts an electrical impulse that travels down wires. When that electrical impulse reaches a second wire, the impulse is transmitted to it and the light bulb goes on. If the electrical circuit is disrupted, the light bulb does not go on. Similarly, in the human body, electrical impulses initiated in your brain travel to the spinal cord and muscle, allowing the muscle to move. In paralysis, part of the electrical circuit in the brain and spinal cord is disrupted. Instead of the light not going on, muscles don't move. Solutions to this problem are being studied in animals with paralysis. In one approach, motor neurons are generated from mouse embryonic stem cells and are surgically placed into the spinal cord of a rat. Specific growth factors are used to generate motor neurons. Other growth factors given into the spinal cord stimulate the surrounding neurons to form connections with the transplanted neurons. Still, other factors stimulate the transplanted motor neurons to send axons out towards muscle. Finally, another factor, called GDNF, given locally near the muscle, results in axons forming active connections with muscle. Thus, the animal has partial improvement in strength and regains the ability to move.
So that's it. That's what all this activity was about. That's the approach we took. And I can show you some of that data, although I really don't want to show you the pictures necessarily. I think the concept of how we attempted to rewire the nervous system was one which is complicated. It's going to take a lot of work, but I think that it does work, or I think it can work. So I'll go over a few things. These are motor neurons. This is looking under a microscope. So these are taking embryonic stem cells, applying those chemicals, and looking under the microscope and seeing these very sophisticated, very motor neuron-like cells that express all of the markers that motor neurons should express. Now, even in a dish, you can see those wires or those electrical cables that we were talking about, that when you put these motor neurons here, and you put the muscle cells on the other side of the dish, they grow, the motor neurons extend their axons or their cables toward the muscle. So they're responding appropriately to the biological cues. Now, this is just to tell you that these are all of the transplantations that we did. There are lots of groups. And you heard about a bunch of chemicals. Some chemicals here, some chemicals there. And we needed to determine which of them or all of them are important in this functional recovery. And so we had to basically do it such that one group got all of it, and all the rest of the groups got all but one of the factors. And we transplanted them all. We blindly kind of put them into cages together and waited 6 months. And we didn't know which was which. And we didn't know animals from one group versus the other. And it was only at the end of the 6-month period that we could break the code and say who recovered. And to cut to the chase, what we found is that 13 of the animals, of the 15 animals in this group, recovered to some degree, but to varying degrees, whereas none of the animals in the other groups did recover. And when we broke the code, it's an amazing moment, because it tells you that you think that this is a biologically relevant way to restore part of this neural circuit, and that's very exciting. It also tells you, though, that you need all of the things we did. It's not simple. It's going to take many things to actually achieve this. Now, I won't go into all of this, because all of these are ways that we scientists say, is there basically anatomic reinnervation? In other words, truthfully, have the motor neurons survived in the spinal cord? Have they themselves been connected by the surrounding environment? Because only if these transplanted cells fit within a neural circuit will they have a functional benefit. Can they extend their wires out to muscle? So what we started to do is we looked in muscle, all the way in the ankle of the rat, and we said, do we see the transplant-derived cables getting there? And we were amazed to find that they do get there. And this, to us, was one of those, oh my goodness, I can't believe what I'm seeing moments. When you see in the muscle, they form the appropriate junctions with the skeletal muscle. So here you have a skeletal muscle that has not seen a neuron in some time. And the rat has been paralyzed for some time. And a new axon comes down the road, stimulated by these various factors, and it gets to the muscle. And what does the muscle do? It forms the appropriate junctions and specializations, and with open arms welcomes that transplanted axon and forms an appropriate junction called a neuromuscular junction with it. So even though the muscle had been quiet for some period of time, it responded to these axons as they made it towards the skeletal muscle. And this will be on the quiz later, exactly what these numbers are, but it tells you that you've got to prove that they're electrically active. And so we actually do nerve conduction studies on these rats. I don't know who's had a nerve conduction study before, but it tests the electrical connectivity of neurons. And if they've gotten there, they better be electrically active, and indeed they are. And the animals get better. Here's the boring way that we scientists like to look at this. If you really want to look at it, though, Chitra's got the videos on her computer. So there will be personal showings afterwards. If you just go on over, and it's a nickel a showing. And so we have concluded. Now, I'll make another point, which is the numbers thing. We effectively generated several hundred new motor units in a paralyzed rat. And these axons migrated over 8 or 9 centimeter distance from the spinal cord of that rat to the ankle of that rat. 200 functioning motor units is really a really small percentage, probably on the order of 3 or 4 or 5% of those numbers that exist in normal unparalyzed individuals or rats. Interestingly, the functional recovery was about 35 to 40%, suggesting that if you just get a couple percent of these motor units to work again, the functional benefit will be in far excess of that. So transplanted embryonic stem cell-derived motor neurons extend to target muscle. As long as we do all this chemical stuff, these things to kind of guide them toward their target, which is skeletal muscle. And although I didn't go into the chemicals, what I would tell you is that we basically recreated development as best we could. when the nervous system is being laid down, when it was being formed in the very beginning, there are a bunch of developmental cues or signposts which guide neurons and tell them what to do and where to go. But once we have finished development of the nervous system, those signposts are taken away. Those cues are no longer needed. The nervous system is now set in place. And what we reasoned and why we use these particular chemicals is we said, well, if we replace them as they were during development, maybe these transplanted cells will respond to the cues and regrow towards skeletal muscle. That's it. Now, all right, thank you. Thank you. Wow. Well, thank you. So I should just say, what will it take from here, right? I mean, because I suspect that might be the first question. Is certain strategies in transplantation will be sooner rather than later. The first things will obviously be, to my mind, activity-based rehab principles, and this is a really dynamic, ongoing relationship that we have with John now. I mean, it really works. People come here and they go over, and it's part of the experience when they come to Johns Hopkins, and John has led the way that the rehab and the activity doesn't just simply allow you to deal with the disability you've got. It changes that disability and it decreases it. So that's the near-term stuff. I think the glial restricted precursors are next, and I think that could be within 2 to 2.5 years. That glial cell transplantation could occur. The motor neuron stuff will not be that quick. And the steps here are as follows. We have to use human embryonic stem cells, and that's an issue. Which human embryonic stem cells do we choose? The human embryonic stem cell that we choose has to be able to do the magical things that these cells have to be able to do all of the reconnections. What if we choose the wrong cells that don't have that ability? What if we're limited in the embryonic stem cell line that we choose and we've done it wrong? What a colossal waste of time and money and hope to go down this road over the next 5 years and to have guessed wrong. And I think what John and I would say is we don't want to guess wrong. We want to get the right embryonic stem cell. We don't want to be limited by some cells that have been derived maybe 10 or 15 years ago. They've been derived in a certain way that is probably not appropriate in transplantation into humans. But if we can get the right line, and we can get the funding to do this, and we can get the okay from the federal government, we will proceed with human embryonic stem cells into larger animals, such as pigs, because the distances that we achieved over 8 centimeters are not the distance that we need to achieve in humans. And many of us believe that that's not the critical hurdle, that once an axon will get into the peripheral nervous system, it will grow as long as the cues are there. But we have to show that. And we have to show above everything else that this approach is safe in animals. And so we've got to do very, very careful toxicology studies in which we show that the transplantation does no harm. So effectiveness in larger animals, safety in larger animals, human embryonic stem cells, get the line right, take that data and go to the FDA and say, we'd like to do this in human patients. And so that's obviously a longer step off. So, questions? Yeah.
What is the recommendation? You
John, you want to talk about that?
Yeah, I mean, I think that, you know, it depends on the condition, but what we were surprised in our study that we did, because we imaged everybody's legs, is 20% of people completely lost, you know, the muscle. I mean, you can't get that back. If it's gone, it's gone. So, but there's a lot of recent work showing that even severe many years out, but we have patients as far as 30 years out, where they're able to get back to what appears to be normal muscle mass. So I think you just need to optimize it. You know, it's like, you know, it's like trying to grow corn in a field that's been sitting in the 100-degree sun, you know, for 2 months. You know, it's going to have difficulty growing. You know, you really need to work to optimize and get the body in that situation to accept these marathons. Activity is a critical part of all this.
And you saw the muscle, right? You saw the MRI that John showed with the muscle after the functional electrical stimulation, where the muscle was more robust and healthy. And that's important because that muscle is now secreting things, I would guess. I'm certain that's the case, right, John? That is secreting growth factors and trophic factors, which would render it better able to accept this advancing axon as it comes. Other questions? Yeah.
What's the glial restricted precursor cell transplant for? What would be the measure of success?
Glial-restricted precursors, what would we measure? It would be in transverse myelitis patients who have, transverse myelitis patients with a single attack initially, a single lesion, and that lesion has contributed disability, part of which is due to demyelination. So the goal is that the transplantation goes into the site of the injury where there are demyelinated axons. The GRPs would remyelinate that area. We would be able to see that by hopefully a functional recovery, but also by an electrical recovery and an MRI recovery. There are ways now to actually image myelin using an MRI. The question I may defer to John is, how do you know if your muscle would be able to accept this transplant?
Yeah, I mean, you know, from imaging alone can tell you if those muscles are there. You can't tell by looking at the leg. You know, obviously, if you have any functional control and movement, spasticity, reflexes, and those legs, you know, okay, so it's no problem. You know, but just because you don't have reflexes and you don't have spasticity doesn't mean you don't have muscles and you don't have nerves to those muscles. That's the biggest misconception in neurology. Once you fall below a threshold, the number of connections that come out is done the same, 5% or something, or some number. You know, you basically don't have function, even though you have functional connections.
Let me repeat what he said. Basically made the point that if you have some movement, then your muscles could be able to respond to this, right? We know that if you have some movement, even if it's incoordinated or weak, or if you have reflexes, that suggests that it's going to be sufficient at some point. He also made the point that if you don't have any of those, it doesn't mean that you can't do it. It just means that there's going to have to be more work to understand and to optimize the muscle for that transplantation. Well, I had one in the back here. Nope, go ahead.
What's Tetris going on?
She'll kill me.
Why only incomplete?
Well, the idea for the glial-restricted precursors is that you must have axons in order to remyelinate, and that in people with a complete injury, this isn't ideal because the axons would be too damaged or not there in order to remyelinate, which doesn't mean that there won't be a therapy. It means that this isn't it, though. Susan? So Susan, for those of you who don't know her, is very, very active in the spinal cord community, has worked very, very hard at the legislative level, is tireless in her efforts to get funding for spinal cord injury and stem cell. And first of all, thank you for all of your efforts. The point she makes is if these things are all okay or approved for other uses, couldn't you use them in combination now or go offshore, go elsewhere? Many or most of these things are approved. What we did is we put this whole cocktail together. And so I think, I get this question a lot, so should people go elsewhere? Well, no. No, because as frustrating as it is, you don't know what you're getting elsewhere. She's going to respond to me. I think she may argue with me. Oh, they want me to go offshore. I may. My patience is wearing thin.
You're not going anywhere.
Other questions? How would my goals be different if I were in California? California has now got $300 million a year per year for the next 10 years for a total of $3 billion in state funding towards stem cell research. That's big. It's been tied up. A lot of it has gone into infrastructure. In other words, new buildings, new complexes, and part of that has to do with the federal regulations. Right? Because if I get federal grants to do legitimate research, and I'd like to do research with non-Bush approved stem cells, I really have to keep those entities completely separate. So although the state funding is helpful, it is still a very cumbersome process to have to keep these entities separate. I want people talking together. I want people thinking the same things. I don't want to keep them separate. It's hard. It's not the optimal environment. Certainly, I think California is a very fertile ground. I think what they've done is great. Maryland has a $15 million state funding allotment for 1 year, although no guarantees that it's going any further than 1 year. All of this kind of patchwork funding, I think, helps. It's not ideal. The regulations will be different in different states. I think that one of the things that we are concerned about as researchers is the ethical foundation for this work. It's got to be done ethically. We think about this all the time. We think about the ethics of it and all the institutional review boards, and the federal government should have a role in that. It should guide this research so that it be done with the proper ethical foundation. And it's not, because it's kind of abdicated its role, and that also concerns us. So, we're all recruited to California on a regular basis, and I'm not going. John, are you going? Other questions? Yep.
Will people with DEVIX be eligible for the glial study?
Will patients with DEVIX disease be eligible for the glial-restricted precursor study? In the first round, no. And again, the goal here is to say we've got to pick a very narrow population up front, and we've got to be right about it. Because if we pick the wrong patient population and show that they don't work, and what if they don't work because we picked the wrong patient population? For example, a patient population who might get a recurrence. Then we would really set everything back. And so I think the goal is to target it in a very narrow way, pick the right population, and then expand it. And that would then follow. Yep.
Right now, what are some of the perceived risk hurdles that might be in?
What would be the perceived risks and hurdles that might come from this glial-restricted precursor?
Or the other stem cell work?
Well, so the glial-restricted precursor study, I guess the risk that everybody would have with a stem cell study is we don't know exactly what the stem cells will do. We've studied them in animals for a long time, and we think they're safe, but like any drug. There are countless numbers of drugs that have been tested in thousands of animals, And the FDA works very hard to minimize or eliminate the risk to humans, but it can't eliminate that risk, and that's going to be the same thing with the stem cells. So there will be a risk as to what these cells do. What's their biology? What do they become? There's a risk of surgery. There's a risk because for a period of time, patients will need to be immunosuppressed, and that scares me. They will need to go on cyclosporin for a period of time, and that concerns me because people get urinary tract infections, and you have to be very, very careful about these things. So this is not to be taken lightly. This is going to be something that is going to be intense. It's going to be scrutinized. It's going to be a lot of follow-up and a lot of uncertainty. John.
So I just wanted to comment on what Doug pointed out, because you guys hear all this, and we hear this in our clinic all the time, and it's relevant to everyone because everyone asks, well, should I go over next year? Should I go over? And I say, well, it takes 2 years to optimize your own spontaneous recovery of function, which it does, and you need to do that before you do that. But although there's positive things happening, it's clear that there's some early sensory return that occurs with a combination of transplantation of almost any cell. But none of those things that are in China are being done as trials, and it's a good example of what happens when the government's not involved, And but the biggest issue that we see with people going over there is the complications not of the cells. In fact, we haven't really seen any complications of cells being transplanted over there, but complications of the surgery. People are coming back with spinal infections, encephalitis, brain injury, seizure disorders that they didn't have before because they developed infections as a result of the surgery, not of the cells. It's because the conditions of the hospitals at many of those places are just not close to what we use here in the United States. And people don't realize that. They assume they're going to the same thing that they found here. And things are moving forward so quickly. What's here today, will be negligible compared to what's here next year and in a year from that. So, I mean, just personally, if put a couple years into optimizing yourself before you go get any stem cell transplantation anywhere in the world because it's not ready. I advocate sometimes for my patients to go get transplants because some people get to the end of the rope and they just got to try it. I understand that, okay, and I'll work through those things. But they got to understand the risks,
Isn't it also the case that if they take the risk to have an implantation like that they take themselves out of the opportunity to be a part of the trial here?
Yeah, it's a good point. I mean, almost any company.
Just repeat it.
Oh, sorry. The question is that you got to realize you may lose something else by going to get that transplant. You may lose your ability to enter into any clinical trial after that, not just a cell transplantation trial, but an exclusion criteria. This is virtually for every trial. Just assume if you get a transplant today, you will never be in a clinical trial again. You will be excluded from every single one. If you're lucky, if there's an exception. Just companies won't take chances when money's involved. They just won't. So that's another thing you lose. So I think that the science is very exciting. There's a bit more work that needs to be done to optimize what we're doing in humans. Right now, transplantation is being done in humans like we do in animals, big operations. There's no need for that. We can do percutaneous transplants that are done in an outpatient center, straight into the spine. These techniques are available. Why put someone through the risks of surgery when it's not necessary and it's extremely costly? Now, when we did our first human transplantation trial, like Doug's talking about, it was done in the big open way too. It was done that way to control things, to understand, but it was done under the most optimal conditions. But in today's world, I think that if you're not doing it as a trial, there's no need to be doing these big operations like you had the first time if you had a spinal cord injury. So those are all the risks and benefits. I mean, obviously, we're super excited about this, and there's something very special about these embryonic stem cells that no other cell has. And I'll just add one point about what Doug said is that the statement that that adult-derived stem cells have been curing people for years, but ESLs haven't is absolutely wrong. There's so much more evidence. ESLs revolutionized science in 1980. That's when mouse embryonic stem cells became available. That's when we could develop transgenic animals. It revolutionized science. That accelerated development of applications. The biggest role of embryonic stem cells, particularly human ESLs, is as a scientific tool of discovery, not as a direct treatment, but to study the response of cells, normal human cells, so you can develop pharmacological therapies and other approaches. So there's been many therapies that have been developed, just not stem cell-based therapies, as a result of stem cells, right? It's accelerated science greater than the Human Genome Project has, mouse ESLs. There's no difference. Human ESLs will do the same. If we don't have a model of a human disease, we can't study it. That's why studying like Alzheimer's disease is so difficult. There's no adequate model. If you don't have a model, how could you study it? How can you develop a treatment? You know, if you can't study normal human cells, particularly in the nervous system, and we don't have any adequate source for this. There's no adequate source. We're using the best you can, you know, and you know, that's why it's just, you know, would we be doing something different if we had $3 billion? Our mission wouldn't be any different, but we'd shift into human ESLs a hell of a lot earlier. I mean, you know, we first started working with human ESLs back in, you know, the latter half of '98, and then we stopped, you know, and really haven't really gotten back into it, you know, because of what's happened. I mean, it's a decade has been lost, but a decade now will translate into therapies being delayed for generations. The indecision in 4 years will affect generations later. That's how accelerated this process is occurring. So the lack of saying, oh, let's just delay it for 8 years. That's, you know, so if a few people get affected, but way more people get affected because things move forward that quickly. Yeah.
We've got the wine and cheese now. Let's stop it, and then we'll be around and we'll ask more questions, and even tomorrow we'll be here as well. I want to thank the Transverse Myelitis Association for this, and